I want to thank you all for coming to this exhibit theater of MLE Genetics Lab. Can everyone hear me? Yeah. So I'm sorry they don't have a wireless mic and I'm short. I'm going to be behind the podium, but I guess you don't want to see me, you want to see the slides, so we should be okay. So I'm going to speaking from here. So what I'm going to show you today is a new assay we have launched for exome sequencing. And those of you who have attended the molecular platform sessions, you did see a little bit of the preview of the assay that has been developed. But at Emory we have made several modifications, and I'm going to go into that and talk a little bit more in depth about how we are also classifying variants. So the outline of my talk today is going to be, I'm going to be talking about whole exome sequencing, the consent options at Emory, the NGS panels versus exome. I think this is like the question that is coming up again and again. What should the physician order and when? The diagnostic year from the panels we have done so far. We have done a lot of panels since 2010. I'm going to show you our data. And a little bit about variant classification and curation and the efforts at Emory. You have already heard a little bit about the ClinGen consortium, to which we are one of the highest contributors to that consortium, and to ClinVar. But I'm going to show you what we have actually done at Emory in terms of recontacting the patients when new data is available. So I'm going to start with the exome boost assay and get into that. So as we all know, single gene testing is pretty well established in the industry now, and it is essentially based on a gene getting reported in the literature, a clinical lab essentially making a decision, and a gene gets launched. The problem with that is that a lot of data gets generated within clinical labs and actually does not get reported outside. So we are collecting a lot of data, it's sitting within the labs, and we're going to come back to this statement towards the end of my talk. But essentially when you're looking at a gene and you're setting up an assay, you're looking at the mutation spectrum of the gene. So at one end of the spectrum is a Duchenne muscular dystrophy gene, DMD gene, which is the largest gene in the human genome. And the majority of the pathogenic events are deletions and duplications, and a minority are point mutations. At the other end of the spectrum, you have the most of the genes which have the majority of the mutations in this gene are actually point mutations. So what we know today is that there are genes where deletion duplications are major events. Then there are some genes where through extensive work from different groups, some common deletion events have been identified, and those are probably the ones you would test first. Then most known genes have a range of 10 to 30% deletions and duplications, and then the newly identified genes we don't know yet. We also know now from the literature and the extensive exome and genome sequencing projects that the complexity of our understanding of genomic disorders is growing. We have many genes, one disorder, many genes, and it's getting complex. We have one gene which can cause autosomal recessive and autosomal dominant disorders as well. And then to add to that mix, the technology has allowed us to get heavily into the cancer area as well for germline and somatic mutations. So I'm going to give you an example of what I mean by this. And I'm using the example of muscular dystrophy because my research is in the area of muscular dystrophy, so I'm very familiar with this area. And from this slide, you can see that there are different subtypes of neuromuscular disorders, but the genes heavily overlap across these different disorders. So the question is that how do you decide which gene to go after? So here's an example. The gene is FKRP, and the range of disorders range from Walker-Warburg syndrome, which is WWS, it's a very severe disorder, to a much milder clinical phenotype, which is LGMD2I. But then you have on the other side FKRP, POMT1, POMT2, and all these range of genes can cause glycosylation disorders and congenital muscular dystrophy. So now you're in a situation where you want to do either one or the other, and how do you decide that? So last year I wrote a review for Advances in Anatomical Pathology for the clinical utility of this new test, where you have a single gene situation, a gene panel situation, and exome and genome sequencing. So just at the outset I wanted to sort of present this slide where we know that in somatic situations in cancer, the hotspot mutation testing or the common mutation testing is probably the highest yield. But that is not so in rare diseases. Of course, there is an exception, cystic fibrosis, we all know that. The targeted panels probably have a higher yield in rare diseases versus cancer, where you don't know how to interpret the data. Then comes exome sequencing and genome sequencing, and with genome sequencing, probably the highest utility has been in the cancer area, where they have found new fusion genes which have helped treatment. So if you look at a comparison between targeted panels, exome and genome, we're going to start with a smaller target region, and when you're doing targeted panels. Exomes, you're trying to interrogate 1 to 2% of the exomes, and the genome is in genome. But what I want to bring your attention to is this particular row here, where the idea here is that when you're doing targeted panels or exome or genome, you want to complete the mutation spectrum of the gene. So that's question number 1. Does the test do that? And then number 2 is that does it cover all the genes, evidence-based review of the genes, and are those genes included in the targeted panel? And what should I order, a panel or an exome for my patient? So if you just look at that in this particular slide, the targeted gene panels which are offered in the industry today are evidence-based gene panels. And what does that essentially mean? That the clinical lab has taken the list of genes, they have read the literature, and that's where you see that some labs would do 10 genes and then the next lab is doing 12 genes. But the point here is that gene truly involved in disease or not? But the things to remember is that when people are doing targeted panels, they complete all the exons of the gene and try to cover the entire mutation spectrum of the gene. Versus exome, we have heard this in the meeting now since yesterday that less than 25% of the genes in the exome are clinically relevant, and it is technically incomplete. Also, it does not cover the entire mutation spectrum of the genes, and I'm going to be talking about that in the OGT exhibit theater later today. So I'm just going to go over the panel flow in our lab. Then what we have tried to do is again address both the questions. We want to cover the entire mutation spectrum of the gene as much as possible. Is trying to get to 98 to 99%. The deep intronic mutations are still going to be there, and nobody can catch them till the time some research labs have reported them. So what we do is that the phenotype data is collected at the outset, and we perform next-gen sequencing. In our validation phase, we know which exons are not going to work because of the technological limitations. So we call it the proactive exon list. We also go and backfill the exons that don't work, but we also have a deletion duplication array, which is we call it targeted gene arrays, to complete the mutation spectrum of the gene. And then we analyze and report the data. So we have today more than 70 gene panels, and we are actually launching a lot more panels. We have an abstract here, one library, many tests, where we have taken all the genes in the SGMD database and created one library. So essentially, even if the gene panel is not launched, it's probably there in the library and can be done as a custom test as well. But just doing the gene panel is not, or with the delute, is not enough. The other thing to remember also is that a lot of testing has also happened on some patients. Some patients have gone through half the test and not completed the rest of it. So the idea here is, can we do the tiered testing approach, where in autism we can do tier 1, which is cytogenetics, and then tier 2 is next generation sequencing. Epilepsy, we have kept the two tests separate, versus short stature is a little bit more complex than that because there are methylation assays involved in it. So let's just look at the how the targeted panel data all comes together. So you are performing the targeted panel. You are going to confirm the variant calls. We are doing the intragenic CNV detection. We are filling in the missing bases. And we are also, whenever a gene has a pseudogene, we have complementary assays, which are Sanger-based assays for the pseudogenes. We take the phenotype data and then also fill in with some other assays. Right now, next-gen technology cannot efficiently do the trinucleotide repeat disorders. So in XLID, we have the FMR1 and the FMR2. We have some biochemical assays. And then, as I said before, there are some genes where there are some common mutation events, such as in the neuromuscular panel, we have the Fukutin large deletion, which needs to be done separately. We have published a lot in this area. I've put the publications down there, but essentially these are about validation of our panels and then talking about a diagnostic year. So moving on to whole exome sequencing. I'm not going to repeat all of this. You have heard this in the meeting today, but essentially we are trying to capture the entire exome, the 22,000 or so genes that are there. It is about 66,000 exons from the SGMD database evaluated at ML Genetics, and we are evaluating about 30,000 variants per individual. What we have chosen to do at MRE is we are doing phenotype-driven analysis, and the data analysis is performed by extracting data for targeted genes. So for that, what it means essentially is that if we are given the information that this patient has a neuromuscular phenotype, then we can actually pull those genes separately and look at them first, and then go on to complete the exome. We look at the carrier status analysis and incidental findings. And as you all know that the carrier status and incidental findings has generated a lot of discussion in the literature. We have ourselves gone through at least 4 versions of the consent form, and we have now a consent form which we think works, and we have got feedback that people actually like it. So the mandatory disclosures on the consent form are the phenotype related genes, that is phenotype, the patient's phenotype, and we are reporting pathogenic, likely pathogenic, and variants of uncertain significance. Under the mandatory disclosures is also the non-phenotype related childhood onset genes, but in this category we are only reporting the pathogenic variants. Then comes the optional disclosures, and even though we have guidelines now which have a list of genes that should be reported on a mandatory basis, we have decided to take a different approach, and that is the opt-in policy, where the patient or the patient's family can choose to receive the results. And these include the carrier status, pathogenic pharmacogenetic variants, non-phenotype related adult onset genes, which is medically actionable, and then medically non-actionable. So going on to the diagnostic years of our gene panels and exome sequencing. We see this range of gene panels which we do, which range from CDG, the congenital muscular dystrophy, limb-girdle muscular dystrophy, neuromuscular panel, eye disorders, autism, and then exomes. And you can see that the highest yield is really in the areas of eye disorders and neuromuscular disorders, where we are hitting more than 60%. Again, you cannot just, the kind of samples that come to the lab, sometimes there is no phenotype details given to us, so probably we can sort that out looking at the phenotype details as well. But the more specific the phenotype, I think the yield is very high. So I think right there we know that if the clinician is able to define the phenotype which is as neuromuscular or eye disorder, it probably makes sense to order a panel. Then the exomes, our yield is right now about 28%, about 0 to 5 incidental findings per patient, and we include the ACMG genes in that. And then you have the pharmaceutical markers and the carrier testing. So this is generally what is going on in most labs, where you have the exome sequencing, the bioinformatics pipeline, and then you do the diagnostic exome carrier screening, and then sometimes what you call the recreational exome. But this is where it gets interesting. And I'm going to move on to this exome boost, or the medical exome, which we have just launched. We know that it is technically incomplete, but what happens when you have done exome sequencing and not detected a pathogenic variant, or in many autosomal recessive conditions, only one pathogenic variant is detected, or in autosomal dominant conditions, nothing is detected, and there could be a de novo variant. You can of course go and do gene discovery, but I think as clinicians, when you get reports back, you're really trying to sort out if there's only one variant detected, a pathogenic variant in autosomal recessive condition, where all the exons filled, was the assay complete? What if there is a deletion or a duplication? How can I get a more complete exome assay? So what we set out to do in collaboration with CHOP and LMM Harvard is to actually try and finish the exome. And the idea here is to cover the technical aspects of the exome, look at the gene and the evidence around the gene and whether it is really clinically related or not, and design a technically complete assay. So the idea of this project is to develop a highly curated gene resource and a technically optimized assay to provide a stepping stone to standardizing the interpretation of genetic variation to fulfill the promise of genomic medicine. So essentially, I actually started this in early 2012 and then joined hands with Birgit Punk at LMM and Avni Santani at CHOP. And we decided that if we get together on this project, we can probably do better. So this is a medical exome project, and the idea here is we set out to design a clinically or medically enhanced kit for genes known to be associated with disease, but we are also developing assays for pseudogenes, genes with pseudogenes for CNV detection, and I'm going to talk later about that in the OGT exhibit theater, repeat regions, and the epigenetic assays as well. So this is some baseline data, and you can very clearly see that between the whole exome, where you have about 5.8% of the regions which are not covered, to if you actually look at a very targeted panel, you still have a significant number of exons or regions which are not covered. And then in between is the 4,600 genes which we thought which were clinically relevant, and the about 4.6% were not covered. So when we actually got together with Agilent and designed the medically enhanced exome assay, we've been doing runs between labs. LMM and CHOP are also running assays, but they have their own panels. Emory has its own panels, and what I can show you is that most of our gene panels are now covered about 99 to 100%. There are quite a few panels which are at 100%. There's not a single exon that is dropping out. The exons which drop out typically are the exons which have pseudogenes associated with it, or exons which are extremely GC rich and we cannot capture them. A good example of that is in the neuromuscular panel, where we have the SCPN1 gene, and the exon 1 of that gene has more than 70% GC. So it's still difficult to capture it. But now that we can complete a panel in an exome test and our dropout is so low, we can easily backfill with standard sequencing. So the idea here is that you start with the phenotype, you look at the complementary assays. By that I mean there's biochemical assays or any additional data that is available to you. Then you're going to decide whether the phenotype is more defined and whether you should be ordering a exome or just a gene panel. If the phenotype is defined as not extreme genetic heterogeneity, you'll probably go and order a panel. There are many single gene assays which we do, and the way our system is set up is that we can do a custom assay for any gene anytime. But if the phenotype is overlapping with some other phenotype, there are multiple phenotypic presentation involved, then you want to do exome. But the thing which is new at Emory now is that you don't need to order an exome which has got gaps in it. What we have done is we are doing the exome boost, which is an enhanced exome with near complete coverage of all medically relevant genes and a very low exon dropout. So that means that, as I said before, we can easily backfill those 1 or 2 or 10 exons which are still dropping out, and at no added cost. So what we are saying now is that essentially we can add a gene panel for free. If you indicate the primary phenotype of your patient, we will make sure that all the exons of that gene are covered, and we'll also do the deletion duplication assay, so you get the complete mutation spectrum of those 50 or 60 genes involved in that phenotypic presentation. So we have a lot of gene targeted arrays in our lab, and what I thought was the next obvious step is that after doing this, if it is still negative, you might want to look at the other genes as well. There are still some gaps, and we admit that the collaboration is still working to develop, to increase the coverage of whatever is still dropping out by developing ancillary assays. But the medical exome array is designed to cover all 4,600 genes on one array. And I'm going to talk about the development of that at the OGT exhibit theater. So that is our exome boost assay, where the gene panel is automatically included with exome sequencing when the phenotype, at least the primary phenotype, is recognizable, and you want to make sure there is no dropout. If after all that the exome is still negative, we are probably looking at a very minority of the exons which are not filled in other genes, or it is probably a new gene. So I think you just get more confidence from doing this kind of an assay. So the question comes again is panels versus exomes. One thing we wanted to focus on, we started doing gene panels in 2010, and we were the first academic lab to get into this, and there are many gene panels and many prices, and we wanted to make sure that it is a little bit more simple for everyone to understand and actually order these tests. So basically, if you look at the comparison of a panel and an exome, we have tried to standardize the cost. So the panels are almost half of the exome. The decision is trying to make it easier so that there is no confusion about, oh, there are 100 genes and what is the cost. So it's all sort of standardized across the board when you're ordering, trying to decide between a panel or an exome. But the important thing to remember is that the gene panel will also include the targeted gene deletion duplication array. Of course, when you're doing gene panels, the number of variants of the significance is going to be a little bit actually higher because you're thoroughly covering every region in the gene. But again, the question is, does the lab have a database where you can evaluate your variants and actually give feedback to the clinicians? So in summary for just this section is that we have the medical exome available at EGL, we have the exome boost option at no added cost. We presented some data to you to distinguish between what to order, whether a panel or an exome. And now I'm going to talk a little bit about reporting and variant classification at Emory. So this is our high-level system diagram. This just shows you our bioinformatics pipeline as a whole. But I want to draw your attention to this particular area here. When we got into exome sequencing and actually next generation sequencing, what we did was we actually took all the 22,000 genes, and at the back end, we have a complete library of primers designed for all these genes. We do single gene sequencing for more than 1,000 genes, but essentially we do get a lot of requests where an exome has been performed somewhere and one variant is reported in autosomal recessive condition, 2 or 3 exons dropped out, and the physicians want those exons completed. So this is a custom assay I'm talking of. I'm going to go and now talk about the MV class program we have available to the physicians. So this is our website, and here you can see this MV class box here. And what we decided to do 2 years ago is, there are a lot of questions that get asked to the lab. Whether we have done the test or not is not important, but we get calls where a test has been performed somewhere. Have we seen it at Emory? Can you tell me what your interpretation is? Because the physician is trying to make a decision. So we went through a big exercise of actually optimizing our variant classification. And this is our own experience, the internal experience. I've tracked it only till 2012. But what it is showing you is that as we launched more and more single genes, the variants of unknown, number of variants of unknown significance also went up. So it was really obvious to us that something like MD class, where we can put our database for a preview to the community, no registration is needed. You just go to the website, click on the box, it takes you to a gene box, you put the gene or the variant you are interested in, and you will know that whether that variant was seen at Emory or not. Now, one thing to remember is we have not pulled every single variant that is out there in databases. These are only variants seen at Emory and classified by Emory. One of the other things is that ACMG requires that when new knowledge is gained on a variant, it should be communicated back to the patient or the physician. So the idea here is, how does the lab do that? Because we are not looking at every variant on a day-to-day basis. We have a database of about 15,000 variants, so it does take time to go through it. So this is what we came up with. If we see a variant today and we reclassify it, our database is set up in such a way that we can scan it backwards and identified the affected patients, it sends an automatic alert to the directors and the genetic counselors. Now, if the classification is from variant of unknown significance to pathogenic, we'll immediately get on it and issue the report. There are many variants that are getting reclassified, and probably we have not been very proactive and from likely benign to benign, but when there is a big change in the classification, we can reissue the report. So this straight scans our database and we can get back to you. But what if you see a variant and you want to know whether MRI has seen it, whether MRI has done an evidence-based review of that variant, and how you can get access to it. So the way to do that is to get on MD class, check if the variant is there. If it is differs from the classification you have on the report issued by MRI or from a different lab, there is a box right next to that variant which is a question mark, and you will send the question back to us and we will get back to you. And there is a poster, and we are also doing chocolate chats to show the impact of MD class. We ourselves have been very surprised. We have got hits from Azerbaijan, and I am just surprised at how many people are accessing MD class. Also, the MRI database is there, a link to the database is there, so many people have actually gone ahead and downloaded the database. We have actively contributed our variants to ClinVar, but we have also contributed to the locus databases, the Leiden database for DMD, where we have sent our variants to them. This approach has been published, and we have given all the data of how we have actually arrived at this program and how we designed it. So this was my last slide. I'm going to stop here. We have about 3 minutes if anyone has any questions.